Hello, everyone, and welcome to today's Keynote webinar titled Point of Care Diagnostics for Infectious Diseases with live Q&A. This webinar is part of the 11th Annual Microbiology Virtual Event Series. My name is Delia Osborne with LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees in our live chat feature during the presentation. You can find live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of our presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the bottom menu on the left portion of your screen to follow the process and obtain your credits. Now let's get started. I now present today's speaker, Dr. Jeanne A. Jordan, Professor Emerita at George Washington University. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Dr. Jordan, you may now begin.
Thank you so much. Appreciate it. Hello, everybody. I am so excited to be here today to talk to you about the new sort of developments in point-of-care testing for infectious diseases. So I have three objectives that I'd like to go through during our talk today. The first would be to review with you the newest update that WHO has on its assured criteria, which they now call reassured. And it's sort of the laundry list of what they would like to see in what they would consider to be the ideal point-of-care test for global use. Next, I'd like to share some of the newest point-of-care testing designs that are being worked on by manufacturers and scientists. And then lastly, but not least of all, I want to be able to go through and spend a little time talking about quality control, quality assurance measures that are absolutely vital for having a accurate point-of-care test result that you're giving out to clinicians and their patients. All right, so to start off with objective one, just last year and less than a year ago, the WHO or World Health Organization updated what they consider to be their ideal test for point-of-care work. And the two newest components are real-time connectivity and ease of specimen collection. So that, in addition to the assured component of the reassured criteria, include having tests that are affordable for global use, having analytical performance characteristics with high sensitivity and specificity, being easy to use, especially for those communities where testing is done out in the field or at home and not done by laboratorians, having a robust and rapid assay so that testing and treating in a clinical setting can be done within the same visit in a timely manner. Having equipment-free point-of-care testing is truly the Achilles heel. This is the hardest, I think, of the criteria to really achieve. And you'll see that as we go through this talk today. And then lastly, deliverable. Once you have manufactured and done all of the sort of validation and verification testing around these point-of-care tests, the idea is now, as an affordable test, how do you get it manufactured where you can serve a large global scale in terms of having enough to be spread across the globe for use. So thinking about the impact of actually creating such an amazing test with these reassured criteria, the impact is truly around test and treat in order to have an accurate and timely diagnosis, so that if one is able to, in a community setting, a mobile van, at home using telehealth connectivity, the ability to have a test result during that visit that can be acted upon in that same day can really be an amazing impact for patient outcomes. And when we think about being out in the field doing surveillance for outbreak response, being able to have real-time data being then submitted through real-time connectivity to laboratories and public health labs, where this data can then be analyzed and policies made and decisions made for with the use of this data. So all of this would really help to impact improving health equity around our globe. So why is it so important to consider deploying global point-of-care tests? And in the sort of space of infectious disease diagnostics, this list of 4 bulleted points really brings home the impact that a global point-of-care test that can be done within minutes or an hour while the patient is being seen is very impactful. I mean, here we have, from just foodborne, waterborne, and airborne pathogens, over 15 million deaths globally each year, according to WHO. In addition, if you think about sexually transmitted infections, the 4 sort of curable ones listed here, Neisseria gonorrhea, chlamydia trachomatis, Trichomonas vaginalis, and Treponema pallidum, the causative agent of syphilis. Here we have 374 million new cases in a year's time in 2020. If one thinks about the fact that many STIs are asymptomatic in individuals, where you have no sort of clinical symptoms, such that without testing and screening, individuals unknowingly can transmit these pathogens to their intimate partners. And so being able to screen for these, even in asymptomatic individuals, can really be powerful for testing and treatment and sort of eliminating that transmission of these pathogens. In addition, we have almost 41 million individuals who are living with HIV, of which last year there was over 1 million cases, new cases of HIV, with over 630,000 deaths each year. And so, again, the ability to have testing for those individuals both living with HIV, to be able to say, okay, my viral load is below the limit of detection, therefore I'm being compliant with my antiviral meds, I'm not at risk of transmitting these infections to others. This is a huge sort of important component, not to mention those individuals who are infected who are unaware of their infection, and so screening could help identify these individuals and thus provide them the opportunity for treatment. And lastly, human papillomavirus, which is a causative agent of many genital cancers, including cervical cancer, anal cancer, penile cancer, and others. There are close to 700,000 new cases of high-risk HPV in a year's time, and over 350,000 deaths annually just from cervical cancer alone. So the ability to use a rapid test for HPV DNA would be very important as a sort of first-line screen for being alerted to monitoring individuals with high-risk HPV for progression to cancers. So on that note, WHO actually sort of modified their recommendation around screening for human papillomavirus based on a study that was published in 2022 from a 3-year study in Papua New Guinea, where they looked at collecting over 4,000 vaginal swabs that were self-collected. These were then run on a molecular platform, the GeneXpert, for high-risk HPV. About 15% were positive for high-risk HPV. 93% of those individuals with an active infection received same-day ablation to remove affected tissue from the cervix. They also went on with this data to sort of compare the molecular approach to the conventional cytology, or basically what we call a pap smear, for detecting high-grade lesions, and found that the molecular approach had very good sensitivity and specificity for predicting high-grade lesions. And so in 2021, WHO decided to switch their recommendation from visual inspection using acetic acid to HPV DNA testing for primary screening for cervical cancer. So that this data helped to sort of drive that change in recommendations. So who uses point-of-care testing? Well, certainly one of the first place one thinks about is in a physician office or an ER in hospitals, where this kind of work has been done for many years. But with the advent of the COVID pandemic, more and more places out in the community took up testing for that and have continued to sort of use this testing even for other infectious diseases. So when we think about urgent care, retail clinics, or pharmacies, people can go in and have rapid tests done for strep throat and other things. And so mobile units have always sort of been out there, especially for STI work or in disaster recovery from even thinking about climate change and very strong, powerful hurricanes and storms, etc., when they're out looking for infectious pathogens. And then again, when we think about self-testing, one comes back to the original at-home self-test, which was the pregnancy test using urine samples. And so this is now a much larger group of different sort of individuals and communities that use point-of-care testing. So if we think about infectious diseases and we want to think about, all right, what would be the most impactful infections to consider designing point-of-care tests for? Certainly one of the first that comes to mind is sexually transmitted infections and the ability to do screening and treating the same day during a visit, because so many of these infections are asymptomatic. So it really helps to prevent transmission. Monitoring viral load for individuals living with HIV, very important, and quantitative point-of-care tests are now being developed such that one can be screened for determining whether you have above the limit of detection and significantly above the limit of detection, to know whether you have a significant viral load that can again be put you at risk for transmitting this pathogen to others. that you're involved with. Antibody detection. This has come up with vaccine use and the idea of being able to sort of study vaccine efficacy, and do people who have been given vaccines, weeks or months later, going back and asking the question, do you have detectable antibody levels? And this can also be done now at a point of care test as opposed to a lab-based test. Drug resistance, when we think about even things like sexually transmitted infections, and specifically Neisseria gonorrhea, where the amount or the percentage of infections that are now being acquired that are antibiotic resistant, this is an important component to include in point of care testing in order to determine or make that decision around which antibiotic or antibiotics am I going to treat this individual who has an active infection. So being able to include drug resistance as part of a point of care test is very, very useful and impactful. And lastly, being able to develop these types of platforms for those infections with high morbidity and mortality. When we think of globally, we think of diarrheal illness in infants and children as having a high morbidity mortality ratio, as well as the severity of infection when you have a central nervous system infection, and then also things like viral hemorrhagic fever. So these are all infections with high morbidity mortality. So any of these that we talked about in this slide would be very impactful in order to have a point of care test to use. So what I'd like to do next is just give you just a small handful of some of the current FDA approved point of care tests that we can consider for examples. And I'll go back to 2010, which seems like a lifetime ago. The first FDA approved assay for hepatitis C virus was an antibody point of care test. And this is was an important sort of test to get out there, because there are a high percentage of individuals that are unaware that they are living with hepatitis C virus. And in the long term, if one does not have treatment, this can put you at risk for liver cancer, and therefore the need for transplantation of that organ in order to survive. And so being able to have a rapid point of care test that can be done within 20 minutes or so to be able to give you that sort of status of, am I antibody positive or not? And then that can allow you to have additional screening and clinical workup and potentially initiation of treatment to cure your hepatitis C virus infection. Now I'm jumping a decade later to 2020, where the FDA gave the emergency use authorization for the first time for a SARS-CoV-2 RNA point of care test. And this, this is an two examples of just a number of different point of care tests for SARS-CoV-2 that came out. Some were lateral flow based, some were nucleic acid amplification based, and I want to introduce a little later on in this talk about isothermal amplification and the CRISPR-Cas technologies that have really innovated and revolutionized, I think. This is kind of the cutting edge of where point of care testing, I think, is going in the near future. Then in 2024, just last year, FDA approved the first CLIA-waived hepatitis C virus RNA point of care test. So this, again, is done on the Gene Expert by Cepheid, and it's a finger prick sample that gives you quantitative RNA levels, so thinking about viral load levels. It's done within an hour and allows for diagnosis of active infection, giving you that viral load, and then allowing that healthcare provider to determine how to initiate and start treatment to eliminate this infection so that you are not at risk years later for developing liver cancer. So the two algorithms that you see here, one is your sort of one-step molecular approach for RNA testing to determine positive versus negative individuals, to then a two-step approach using first the antibody point-of-care test, and then knowing sort of those, which individuals, the ones testing positive for antibody, would then go on to do a molecular approach. Again, all of this could be done within about less than an hour and a half within a visit time frame, where you could then determine of the antibody positives which have positive viral levels as well for initiating treatment. So obviously a lateral flow antibody test, very rapid, very inexpensive compared to a molecular approach, but both of these are very sort of important assays, and both of them have important uses in the clinical scheme of diagnosing hepatitis C virus. Then this year, the FDA approved a sample collection device called a teal wand, and it's for home collection of a vaginal sample for high-risk HPV DNA screening for cervical cancer. We have been involved at GW in a small trial using collection devices like these for individual women living with HIV who are at high risk for cervical cancer, for self-collection at home in the comfort and privacy of your own home. And then it's mailed in, and then the samples are then tested within the laboratory, just as this. And so in addition to that, this system also allows one to have, say, a telehealth medicine sort of connectivity over a computer or telephone, so that you can have a virtual follow-up or an in-person visit in order to be provided with those results and a health plan if one needs to have follow-up testing for cervical cancer screening. Okay, so if we think about point-of-care testing, they can be designed to detect these three main sort of flavors of a pathogen. You can either look for antigens, specific proteins that are that have high level of specificity for the pathogen you're looking to detect. You can look for antibodies to specific pathogens. And lastly, nucleic acids. So these are sort of the three different types of targets that you have to sit down when you are thinking about putting together a point-of-care diagnostic test to say which of these three, or more than one, would you like to consider. And sometimes both antigen and antibodies are designed and built into the same lateral flow test, and I have an example of that I'll show you in a minute. So samples collected for point-of-care testing, this list has certainly grown from urine for pregnancy testing to then finger prick for potentially HIV point-of-care testing on a lateral flow device for the antibody and or antigen, to then many respiratory type samples for respiratory pathogens. This got elevated during the COVID pandemic, when one wanted to be able to do community screening or at-home testing to determine whether you had an active infection. And then others as well, certainly for sexually transmitted infections and cervical cancer screening, etc. Genital samples have been a very popular one to include for point-of-care testing. So what platform should we use for point-of-care testing? So the two main ones that I think about, and there are certainly others, but the most common one is your lateral flow immunoassay. And these are usually instrument-free, so that equipment-free criteria that is part of the WHO reassured Achilles heel. Many of these lateral flow tests truly allow you to have test results without the need of an expensive piece of equipment. And again, these lateral flow immunoassays can detect any of the three: antigens, antibodies, nucleic acid, and sometimes there's an upfront amplification step for nucleic acid. Sometimes there's nucleic acid extraction that's required. Sometimes it's just a crude cell lysate, but there are many different sort of ways to utilize this particular platform. The other is to target nucleic acid specifically, being the one that, as we all know, when we sort of moved into that era of doing molecular-based testing like PCR, polymerase chain reaction assays, for STIs, the first couple that came out from Roche were Neisseria gonorrhea and Chlamydia trachomatis. These use nucleic acid as their target to specifically detect these pathogens. So thermocycling was sort of the first nucleic acid amplification application to be used. And then but that requires thermocyclers, which are not inexpensive. So when we're thinking about the reassured criteria from WHO, the thought of, oh, how can we eliminate thermocyclers? The idea of using a single temperature or isothermal amplification is something that is now sort of a common thought and was developed quite a few years ago by scientists who used multiple primers for the loop amplification mediated polymerase, so LAMP assays. But there are others that are also out there as well that can do amplification of a target at a single temperature. So this allows one to think about, oh, all I really need is a heat block or a water bath set at a specific temperature. And those are pretty inexpensive pieces of equipment that one needs. But again, those require electricity. And when we think about this as a global sort of need, not all places have reliable electricity, especially if you're out in the field doing community-based work. Okay, so I'm going to go through a little bit on sort of isothermal amplification as a point-of-care test as it relates to its sort of marriage with a detection component that is the CRISPR-Cas technology. So if we think about the amplification components from the beginning, one has to collect a sample, and then there's some kind of processing of that sample that needs to occur. And if you're able to, it's best if one can get away with using a crude cell lysate. Here you're using some sort of detergent in order to disrupt membranes that allow for liberation of the target nucleic acid that you're looking to amplify. If you aren't successful enough to be able to use a crude cell lysate, nucleic acid extraction is also a option. But this again requires sort of electricity and microfluidics in a cartridge, and that is usually a more expensive piece of instrumentation that one uses in order to extract and have purified nucleic acid to use as your target. So the next step is then, okay, let's design the primers we need in order to do our amplification at a single temperature for isothermal amplification. And then lastly, all right, you've made all these amplicons. How are you going to detect them? And there are many different ways that the LAMP sort of platform uses. One can use fluorescence if you have a fluorophore labeled probe that you're using, or but that requires a fluorometer, right, or some device in order to read the light emissions after excitation. So one can also use colorimetric or turbometric options where you are looking for a color change or for the development of a turbid sort of, think about latex agglutination where you've got turbidity occurring because you've got white particles forming. So these can be read by eye, do not need any kind of instrumentation. All right, if we think about the sort of CRISPR-Cas-based technology, this is a relatively new technology that has really gotten investigators and manufacturers excited about this very sensitive and specific technology that can be done. So here you've got your target sort of recognition where there is, your target is hybridized to a specific nucleic acid. And this is then designed, and I'll show you in the next slide, sort of a cartoon of what I'm talking about. This allows for sort of the 3 and 5 prime ends of this sort of nucleic acid strand to hybridize to your target, which then allows for DNA ligase enzyme to join these together to form a circle. And then with your sort of rolling circle amplification polymerases in the mix, you're able to make just a ton of these amplicons that are then detected using the CRISPR-Cas-base non-specific transactivated cleavage. And so we'll go to the next couple of slides will help us out in terms of thinking about what this actually looks like. So if you have your target here, and then your sort of nucleic acid that you've engineered to have complementarity to the 3 and 5 prime ends of your target, and then the DNA ligase, which closes this circle, you now have this substrate that with rolling circle amplification and polymerases can then generate a huge amount of amplicons again at a single temperature. And with this sort of system, this next component is where your CRISPR-Cas, and in this specific example, Cas12a, this particular enzyme has nuclease activity, and it can cleave single-stranded nucleic acid. And so once this sort of guide RNA or DNA binds to the specific target that we showed in the last slide, and you have this amplification, and there are many different ways to achieve this, but in the last slide, our example was this rolling circle amplification. You have all of these amplicons. This then activates this particular Cas12 enzyme to cleave reporter molecules. And this is an indiscriminate cleavage. So as opposed to the cleavage here, which is target specific. This cleavage down here, which is sort of the secondary activation of this enzyme, allows for cleavage of non-specific targets. And so what you now have in this mix are doubly labeled probes that can be cleaved and the fluorescence emitted with excitation and emission. And so the positive reaction of this very complicated sort of system can allow you to visualize. And in this case, it's a black and white photo, but these usually are sort of using blue light. You have these sort of green fluorescent signals that are visualized using a lamp to look for positive or negative reactions. And so this allows for isothermal amplification and detection, and the sensitivity of these CRISPR-Cas-based systems are really, really good, even down to a few copies per ml for different pathogenic targets that have been published in the literature in recent years. So let's turn our attention now to lateral flow strips, and what are they? Like, how are they made and what do they do? So if we think about this sort of equipment-free component, we start out on the left side of this lateral flow device with the sample pad. And this is an absorbent piece of material that you put your sample, be it a few drops of blood or a few drops of urine or saliva or part of a respiratory sample, etc., on that pad. And then there is always what they call a chase buffer that is then added immediately after you add your specimen. And this sort of initiates this flow of fluid. And the flow is driven by the right-hand end of this lateral flow strip, which is an absorbent pad that drives this capillary action. So this fluid, the chase buffer in the sample, first hits this conjugate pad. That has all of your chemistry in it. And so if you are looking to detect, say, an antigen, in this pad there's going to be a labeled antibody present that it can have the option or ability to bind or hybridize to. And then it goes on to the nitrocellulose strip, where reagents have been specifically laid down in a linear fraction as lines, as a test line for what you're looking to detect, and then a control line, which is your internal control, which is absolutely necessary for one to have a result that you have trust in its results, so that the internal control line must always turn positive, especially in a negative test result. So that if you have a positive control line here and nothing detected on your test line, you can have assurance that you have a true negative and not a false negative. And so building in internal controls is absolutely critical. So this is sort of all the different sort of pieces of absorbent paper, nitrocellulose paper, the chemistry here. This is what a lateral flow strip kind of looks like at a very close look. So this is an example of the Alere antigen antibody point-of-care test for HIV-1, where it is obviously a instrument-free detection of both HIV antigen and antibody in the same test, along with an internal control. So there's a line for antibody here, and then a line for antigen here for HIV, and then the internal control, which it assesses that the chemistry, the substrate and enzymes and all the chemistry is working. So These 4 examples happen to show valid internal controls for all 4 strips and positive visual lines for antigen for HIV, but no detection of antibody. And this came from a tissue culture system where I was growing HE5 cells, which produce P24 antigen, but obviously no antibody. So if we think about comparing rapid antigen lateral flow immunoassays, which we just had an example of, with a nucleic acid point-of-care test, and think about, all right, how well do each of these perform for sensitivity and specificity for a handful of different pathogens, you can see that on average, specificities are pretty comparable, but it's the sensitivities that have improved when one is looking to use a nucleic acid amplification assay. So this is not a nucleic acid test that tests directly for nucleic acid without amplification, but with amplification, be that isothermal or a PCR thermocycling type assay. So it's not surprising that the point-of-care test using a amplification-based test for nucleic acid would have increased sensitivity over an antigen-based non-amplified assay. All right, so in the last few years, there have been other options for detecting targets that I just kind of want to go over so that you have a tiny appreciation for some of these. So the engineering folks in the world have come up with these nanoparticle detection-based lateral flow assays that help to improve the sensitivity compared to that last table that we just looked at a couple slides ago. So in this case, they're using a gold nanoparticle that the antibody is conjugated to. That is then part of the chemistry for this lateral flow device. So the sample is being added, the chase buffer is put on, and one is looking for antigen for this particular pathogen that you're developing the assay for. It can agglutinate with or combine with the antibody that's conjugated to the gold nanoparticle. And if there are enough of these agglutination reactions or complexes formed, there is a color change that occurs. And that's physics beyond what I can explain to you right now. But the color will change, and this can give you that sort of positive result or negative result. And this can improve the sensitivity based on using these gold nanoparticles. Another option is to use quantum dot fluorescence detection. Again, if you're looking for a specific antigen, you would have an antibody conjugated to these quantum dots, and you would, with enough agglutination or antigen antibody binding, show a luminescence or fluorescence that occurs. And again, it's a great way to sort of increase the sensitivity of a lateral flow assay for antigen detection without any sort of amplification of the target like you have in thermocycling or isothermal amplification of nucleic acid. One of the downsides to quantum dots is that there is a certain level of autofluorescence that can't be solved, and so that it can lead to higher backgrounds and potentially false positives. And so there has to be work around where you sort of set that cutoff for background fluorescence in order to avoid those false positive results. The other sort of weakness or downside to this is that quantum dots require a cold chain storage. So one needs refrigeration, and that requires electricity oftentimes, right? So it also needs a handheld device for emission and excitation and emission of these of the fluorescence. And so this does not eliminate the need for equipment for point-of-care testing. And the last is a magnetic nanoparticle immunoassay, where scientists and engineers have found that these magnetic nanoparticles have an endogenous peroxidase activity. And if you sort of utilize that enzymatic activity with hydrogen peroxide as a substrate, one can, with sort of this antigen nanoparticle sort of immunoassay, allow for oxygen bubble formation, because peroxidase breaks down hydrogen peroxide into water and oxygen. And so bubble formation can occur. And you can either measure the change in resistance by oxygen formation in a sort of microfluidic device, or one can use a colorimetric substrate like TMB, where you're visualizing a color change. And so these are also new sort of nanoparticles that one can build into immunoassays for lateral flow devices. So some of the other designs that have sort of advanced the field of point-of-care testing, microfluidics has been a huge one, where you can miniaturize the device that you are using for your test device. And this, again, is not a lateral flow immunoassay, but this is more a cartridge-based lab-on-a-chip device, where it, because it is scaled down, the amount of reagents needed, the amount of sample needed are all reduced, and so it reduces the cost of the chemistry of the device, etc., and uses very low energy consumption, as well as having high sensitivity. Lyophilized reagents has been another godsend, where one of the first companies out there using lyophilized agents for molecular based amplification testing was Cepheid with their Gene Expert, and even before that with some of their other instruments, where you can have dried down reagents that do not need cold storage. And so storage at room temperature within sealed bags that keep the humidity out so that they stay desiccated. These can stay stable for long amounts of time at room temperature, even sort of summertime room temperatures. And so it eliminates that cold chain storage. And then the last, which is newer to come out, is communability, which is sort of your use of a smartphone for being able to send that information or data through the phone to clinicians that are not where the testing is being done, or epidemiologists who are trying to analyze data for surveillance work, etc. So those have all sort of really helped to advance these point-of-care tests. All right, the last of my objectives is to spend a couple of minutes talking about requirements for point-of-care testing. So one thinks about lateral flow devices especially as being very simple to use. You drop a few things of sample on, a few drops of your chase buffer, and away we go. Well, there's a lot of work behind keeping those tests valid, the results documented, the quality control, quality assurance components. And so I want to spend a few minutes making sure you appreciate the work that goes on behind that for these tests to be considered valid and accurate and trustworthy. So for waived testing, where CLIA has defined a test as being waived, is a simple sort of laboratory examination and procedure that have insignificant risk with an erroneous result. So these are low-risk sorts of results, but obviously very impactful. So these waived testings require CLIA certificates, and so that is an application that has to be applied for, and you are given permission and this certificate from your local state or public health agency, and you are issued a certificate of waiver for you to actually legitimately do this testing for individuals. And these can be unannounced visits that where you have to show your certificate of waiver and show all of the other documentation as well for how you've been doing this testing. So team competency to perform these point-of-care tests is vital, because if we think about these tests being done out in the field at urgent care, walk-in pharmacies, etc., most of these people doing the testing are non-laboratorian, so they've not been steeped in the culture of quality control, quality assurance as a laboratorian. And so it's vital for you to train these individuals to have an appreciation of how to troubleshoot assays when things are not working, how to adhere to the manufacturing instructions in your standard operating procedure that you've written out, and compliance with all of the quality management standards that you need to fulfill in order to be compliant around point-of-care testing. So there are many challenges to point-of-care testing, and certainly testing errors and ensuring accuracy and reliability. These are all things that have to be addressed when one is rolling out point-of-care testing, especially in a non-laboratory setting. So I could not find anything past 2008, but this is the sort of 8 of the top 10 CLIA point-of-care deficiencies that are cited when one has a CLIA inspection for point-of-care testing. So the top here are reagents are not stored properly. Some of them may need to be at refrigerator temperatures and may be stored at room temperatures. Use of expired reagents. This is a huge one. People are like, I don't want to throw away these unused tests. This is like money being thrown in the garbage, right? But you cannot use expired reagents and be assured that your test result is valid. Lack of proficiency testing. When one is doing this kind of testing, you need to involve yourself in quarterly proficiency testing where samples are sent to you by an agency and you test them, you send back the results, and you get your sort of report card as to how well you did. Having an assessment or corrective action built into your standard operating procedures is key in order to keep track of issues that arise and how you correct those deficiencies or issues that arise. Not having complete standard operating procedures, not following the directions for these SOPs, or if there's equipment involved, not doing equipment maintenance to make sure that the equipment is working as it should. And then obviously inadequate or missing information. So quality assurance, quality control, I know I've beaten this to the ground, is never a one and done. It is an ongoing labor of love that one has to commit to. So when you think about assessing point-of-care accuracy, there are, when you involve yourself in quarterly proficiency testing, you have the option of going out and searching for quality control material that can be used for assessing new lot numbers of these kits that come in. And kits will oftentimes have internal positive and negative controls that you can use. But it's also important to think about spending the money for a third-party vendor for these quality control materials, because they're objective and independent and can help you determine whether or not this kit is functioning at the level that it should be functioning at compared to the internal controls that may also be in the kit. So really important to minimize the risk of releasing inaccurate test results. So in terms of thinking about assay verification, once you have made that decision that you're going to bring a new point-of-care test online, you've gotten a certificate of waiver from CLIA to do this, you're now at a point where you need to assess this new kit using samples of known results in order to establish that you, as the individuals doing the testing, can actually perform as intended. And so all of this is critical for having the confidence to move forward. And then again, all of these sort of it's not a one and done, but an ongoing quality control, quality assurance process that needs to do. So when we think about point-of-care testing, the strengths are that we've seen that nucleic acid amplification testing is more sensitive than antigen-based, and that there are more and more samples that can be tested. The weaknesses certainly include the instrumentation and kit cost for things that are doing nucleic acid amplification testing, and the quality that one needs to keep track of for test performance with non-lab staff. Opportunities are really being able to get these tests out into the community to help increase testing and surveillance and treatment if needed. AI, I think, is going to help with sort of interpreting point-of-care test results, which may be informative, as well as the use of smartphones with the GPS and built-in cameras that are already being used. Threats, certainly regulatory issues and the delivery model being challenged of having all testing being done in the lab. That's just not going to happen anymore. So I'd like to end with this question around where should non-waived NAT point-of-care tests be performed? And the American Academy of Microbiology came out with a position statement to say that near-patient point-of-care work for nucleic acid amplification non-waived point-of-care tests should really have oversight by laboratory-based personnel. So that doesn't mean the lab personnel has to be out into the community, but there should be oversight, in their opinion, for this. So I will end with that. And there's a list of a few references that were used for this presentation that really were great review articles, etc. So I appreciate your time and your attention.
Thank you so much, Dr. Jordan. We are going to start our Q&A portion of the webinar. I know we're a little short on time, but we are going to get to as many questions as we can. So if you have a question you'd like to ask, please click on the Ask a Question box located just below the slide viewing area. We're going to get started here with our first question. What does hepatitis B virus have that hepatitis C virus does not, that allows for it to have a vaccine available? Could some of the components that of the hep B just be tweaked to make hep C vaccine?
Okay, thank you for that question. These are two totally different viruses, and the hep C virus is an RNA virus, and I believe hep B is a DNA virus. So again, thinking about the higher level of mutation, etc., in RNA viruses with shift and drift, etc., compared to DNA viruses. they are just two totally different categories of viruses, and it's always been easier to make vaccines against DNA pathogens than RNA pathogens. That's probably not the complete answer that you were looking for, but that's all I can think of right now to help you out with that.
Thank you very much for that. We're going to try to squeeze one more in here. How can identify for gold nanoparticles based AGAB DNA fluorescent detection strips showing difference in cervical cancer swabs for early HIV detection owing to heterogeneity, in terms of biomarkers? Apologies, I did not help with the mispronunciation of that word.
Okay, so they're looking to say, can you use the gold nanoparticles for, okay, so biomarkers for cervical cancer and tests for detecting the pathogen itself are two separate components. So all of these assays that are out there now detect just high-risk HPV, and they do not detect biomarkers. And that's really that level that NIH is trying to fund grants to identify these particular biomarkers, be they we just finished a 5-year R01 looking at biomarkers for better predicting anal cancer, and looked at methylation and looked at some other epigenetic markers. And so that's sort of the field where, yes, many people will have HPV infections and never go on to have risk of progression to cervical cancer or anal cancer. And it's like, what's the biomarker that I'm looking for? So that's the next sort of level of being able to better predict risk of cell dysplasia. And that's really what's needed. And certainly gold nanoparticles could be used bound to antibodies that react to specific antigens that have been identified as biomarkers for being able to predict high-grade lesions. So this would be something that I think manufacturers and scientists should be out working on now, because this is a very important question.
Wonderful. Well, thank you so much. And thank you for taking the time to answer that one. Thank you for your important research and your presentation. Before we go, I would like to thank our audience for joining us today and for their very interesting questions. Any questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via contact information that was provided at the time of registration. Additionally, this webcast can be viewed on demand and CE credits available for up to 1 year. We encourage you to share the on-demand version of this presentation with any colleagues who may have missed today's live event. Until next time, take care, everyone. Enjoy the rest of the live event. Bye-bye.
Thank you so much.